Let's see now the vision. For the vision, it involves detecting and interpretation or interpreting visual stimuli by converting light energy into nerve impulses and transmitting them to the brain. Now, of course, as you know, we have the eye as the organ that is responsible for this process. And we have several structures within the eye. We have the sclera. This is the connective tissue surrounding and protecting the eye. The cornea is an extension of the sclera, transparent in this case. The sclera is the white portion of the eye that we are seeing. The cornea is transparent to allow the light to pass through. We have the aqueous humor, which is a type of a fluid behind the cornea. We have the iris, that is the colored part of our eyes. We have the lens. Then we have the vitreous humor. This is another type of fluid inside our eye. Then we have the retina. This is the neural layer that is stimulated and responding to the eye, to the light. We have the optic nerve that is responsible for transmitting these signals to the brain. And within the retina, we have two areas. We have the macula, which is the area for focus imaging or focus looking. And we have the optic disc, which is the area where the optic nerve is exiting the eye. So these are in general different structures associated with the eye. Each one of them in general has different function as they are summarized by this table. Now, for the amount of eye or regulating the amount of eye entering, regulating the amount of light and focusing the image, we have two major, or maybe two or three major structures, depends how you are looking at them. Now, for regulating the amount of eye, we have the iris. This is the iris. The iris basically is attached to smooth muscles, and you can think of it as a smooth muscle itself, that causing the pupil. The pupil is the area, the opening between the iris, causing the pupil to either constrict or dilate to control the amount of light getting into the eye. To focus, it involves also bending of light through the cornea and the lens. So the image is always falling into the retina. And when we have a close object, the lens are going to adjust, meaning that they are going to have more bending, more curvatures to enable that image to focus or to fall down on the retina. Whereas for the distant objects, the lens are going to be stretched a little bit by the muscles, the ciliary muscles. These are also a group of smooth muscles. So again, the same concept, the image would fall directly on the retina. Now, as a human, our ability to accommodate for close and far or distant objects or close and distant looking would downgraded, I would say, as we are progressing in age. That's why as we are getting older, we need to wear glasses for either close objects or distant objects. And usually it is more for close objects, the reading glasses. Now, eye shape would affect the focus, because the normal eye, always the light or the image is going always to fall on the retina. However, we have conditions like the myopia, which is nearsightedness. Here, the persons can see near objects clearly, but distant objects they would fall out of focus, simply because the focal points in this case would fall in front of the retina. So on the retina, the retina would receive a blurry image, and this would be corrected through concave lens. The other form is the hyperopia, which is the farsightedness. In this case, person can see distant objects clearly, but the near objects are out of focus. Why? Because the near objects are going to fall behind the retina, and this will be corrected through convex lens. The other form of abnormalities in eye is something called astigmatism, which is due to abnormal curvatures of either the cornea or the lens. So there are some abnormalities on the way the cornea is curved or the lens are curved. So we will have different images falling on different focal points on the retina, and this will be corrected through a special lens. Now, to understand vision, we need to understand that light is converted to action potentials, and this is happening within the retina. Within the retina, or the retina itself, allows us to see in color, adapt to varying light intensities, and receive images. And here within the retina, we have several layers. We have the outermost layer, which is pigmented cells, and these are going to absorb excessive amount of light, or the light that is excessive. Then we have the second layer, which are the photoreceptors. These are the photoreceptors, and we have two types, rods and cones, and we'll talk a little bit about them. Then we have bipolar cells, and we will see what do they do. And then the last layer, which is the innermost layer, the ganglion cells. Now, the action potential, we said in order to see light need to be converted into action potential. That is the function of the photoreceptor cells. These are going to convert the light into electrical impulses. They transmit them to the bipolar cells, and from the bipolar cells, the impulses would be transmitted to the ganglion cells. Ganglion cells, they have long axons. They are forming the optic nerve. So through the axons of the ganglion cells, these electrical impulses will be transmitted to the brain. For the photoreceptors, we said we have rods and cones. And for these rods, this is like coming from their shape. These are the cones, these are the rods. Now both would respond to light because they have photopigments, and these photopigments are somehow sensitive to light, although they are in different sensitivity. We have more rods than cones, and both are important in vision, although one of them, the rods, are going to work only in dim light. So these are very sensitive to light. I'll write it. So these are for dim light or low light vision, whereas the cones for bright light. So cones would work also with color. They will enable us to see color images, whereas rods, no, for dim light and no color. Technically, at any certain condition of lighting, unless it's extremely low light, both of them you can say they are working. Although, again, in a bright light, it's mainly cone cells, whereas in dim light or low light condition, it is the rod cells. Now, rods and cones are not evenly distributed. in the retina. We have the cones are mainly within the macula, the area of high focus or detailed imaging, whereas along the retina we have more of these rods. For the cones, actually I forgot to mention the name of the pigment that is called rhodopsin. These are the photopigments within the rods, and these are more sensitive to light than the photopigments in the cones. Now, for the cones, we said these are responsible for our color vision. We have three types of cones: red, green, blue. Each one would be stimulated to certain wavelength corresponding to one of the three different colors. And the brain would intercept the ratio of impulses coming from the ganglion cells connected to the three kinds of cones, and that's how we are going to see colors. As we said, cones require stronger light, so they would be working in a bright light, whereas in dim light we use rods and we don't see colors. And as I said, cones are responsible for the detailed image or the visual acuity of our vision. Now, for the visual receptor adaptation, it depends on the adjustment of the pupil size by iris and the adaptation of rods. What we are talking about is how do we feel or how do we see if we move from light to dark, or the other way around, from dark to light. So these are called the visual receptors adaptation. And adaptation takes longer time when going from bright light to dim light, because the bright light uses up the rhodopsin temporarily, and it will take several minutes to remake it. Rhodopsin, again, is very sensitive to light. It will break down upon sudden light or upon bright light, and it will take several minutes to visualize it. That is why when we move from bright light to dim light, we will see darkness. So if you are in the living room and you are going, say for example, into the bedroom and you don't want to turn on the light, basically for a couple of minutes you will see nothing. It's just black. This is a video about the human eye. It explains some of the concepts that we covered. And very few last slides. Disorders of the eye, of ears and hearing. We have the deafness, of course, losing of hearing. We have otitis media, inflammation of the middle ear. We have Meniere syndrome, which is a condition in the inner ear that impairs hearing and balance. For the eye and vision, we have retinal detachment. The retina is separated from the choroid, the layer beneath it. In this case, patients will see flashes of light, blurred vision, loss of peripheral vision. We have cataract, when the lens become oblique, non-transparent. Usually would be replaced with artificial lens. And we have glaucoma, when the pressure inside the eye raises due improper draining of aqueous humor. This usually can be treated with drugs or even surgery. If untreated, it might lead to blindness. As you can see, this is an example of cataract, and this is an example of the attached retina. Also, we have some age-related macular degeneration. The retina will detach and photoreceptors in the macula will degenerate. So we will lose visual acuity, the ability to see in details. And this is the leading cause of blindness in people over 60. Also, we will have the color blindness when we have lack of one of the three types, of one or more of the three types of cones. In this case, patients would be unable to distinguish the full range of colors. Usually, the red-green color blindness is often inherited. These are different types of visions. This is normal vision. This is with the glaucoma. This is the macular degeneration. As you can see, you can't see the focus of the image. And this is a paper to test the color vision. Usually you will have different sets of papers with different colors and different numbers, and usually patients need to identify the numbers in the middle or the letters in the middle to determine if they have color blindness and what type of color blindness. I believe that's all for this chapter. Please post your comments, questions, thoughts in the forum, and we will talk more in the lab.